Good morning, everyone. Thank you for attending today's webinar, which is support for MiSeq data in Avadis NGS. Avadis NGS is an NGS product which has been out in the market for more than 1 year now. The goal of the product is to allow biologists to carry out sophisticated NGS analysis in a user-friendly and interactive setting. The product is developed on the same underlying platform as GeneSpring. Those of you who already are GeneSpring customers might find the user interface very similar and intuitive. The product right from its first version supported import of aligned files which are in SAM, BAM or ELAN format. After the files are imported into the tool, one can carry out ChIP-seq, RNA-seq or DNA-seq analysis. The latest version of Avadis NGS 1.3, which was released this month, contains support for small RNA analysis. It also includes support for small RNA and DNA read alignment using a cobweb aligner which is incorporated into the product. Today's webinar is the extra support that we provide for analyzing data which has been generated out of the MiSeq platform. So some of the applications of the MiSeq benchtop sequencer are resequencing, where we carry out high coverage sequencing of maybe a small known genome. A second main application is Amplicon sequencing, where targeted regions of a large number of samples are covered at high depth. Then we have small RNA analysis and metagenomics, where we assess the profile of different bacterial species in a natural sample and verify how the profile changes in different samples. And finally, de novo assembly of small organism genomes is also one of the MiSeq applications. Of these 5 application areas, the first 3 application areas, resequencing, Amplicon sequencing, and small RNA analysis, are provided comprehensive support within the platform. We have also carried out some internal experiments on how to go about supporting metagenomics and de novo assembly data also. Those results I'll present towards the end of the webinar. So on this slide, we have the standard NGS data analysis pipeline. The sequencer generates reads, typically in FASTA-Q format. The reads may then be aligned against a reference genome, and the output may be either in a custom format such as ELAN format, or it could be in standard SAM-BAM formats. Some of the sequencers also include additional application-specific software, whereby they can detect the variants with regards to a reference genome and output the DNA variants in the form of VCF files. Or in the case of RNA-seq or small RNA-seq experiments, they may analyze the alignment across the genome in the context of annotations and generate a quantification result which is in the form of a TSV file. Avadis NGS allows you to import many of these results into the tool. If all of this data has been generated via a MiSeq run, we can directly import the variants that have been generated by MiSeq. We can also import the tab delimited quantification results. If one wishes to carry out the analysis within Avadis NGS itself, one can import the aligned reads which are in SAM-BAM format into the tool and carry out the SNP detection or the quantification within the tool. Furthermore, if people want to start with the alignment itself, they can take the FASTA-Q files, import them into the alignment part of the tool, carry out the alignment against the reference genome before carrying out the SNP detection or quantification steps. So as you can see, data can be imported from different parts of this particular pipeline, and what we'll do now is to describe the different MiSeq applications. What is the data that is generated by them by the MiSeq run, that is the software included with the MiSeq sequencer. Which of those data can be imported into Avadis NGS? What are the MiSeq specific import features which allow for a seamless integration of the MiSeq run and Avadis NGS, so you can use the best of both worlds? And after the data has been imported, how can the downstream analysis be carried out on the data? So let's start by taking a look at resequencing. The output of a MiSeq run is a folder. In this particular case, the folder name is 110509 bold, etc. All of the data sets that I'll be showing in today's presentation were made available by the MiSeq product team in August of 2001. They're used here only for demonstrative purposes, and if you have a MiSeq instrument which came out after the date, the results might be different. The folder structure has a deep hierarchy. As you'll notice out here, underneath the resequencing folder, you have a data subdirectory which internally contains intensities, which in turn contains base calls and alignment, etc. And each of these folders has a large number of files. So while the files might still be in a format which can be imported into Avadis NGS, that is, they could be BAM or SAM files, they could be VCF format files. Finding the files inside this nested hierarchy is not a trivial matter. What we need to do to carry out resequencing analysis is to identify the sample information, the aligned files, and the SNP and indel results, which can be imported into Avadis NGS. If you take a look inside the alignment directory, which contains most of these files, you'll see that there are a lot of files out here. We have reads which have been aligned against the individual chromosomes and the plasmids of the bacterial organism in question. We have SNP and indel results, etc. So we have the aligned read files in BAM format. We have the indel and SNP results in VCF format, and the sample name itself is present in a different sheet elsewhere in the hierarchy. To import all of this data into Avadis NGS, we can start by creating a new resequencing experiment. Internally, this means that we create an experiment of type DNA variant analysis. The resequencing was against the Rhodobacter sphaeroides organism. The annotations for this organism are available on the Avadis NGS annotation server, and you can directly download them through the annotation manager in the tool. After that, we specify the library layout, which is paired in. Then we are prompted for loading the MiSeq samples. Avadis NGS 1.3 includes a new MiSeq button out here, which allows you to load results from the run folder into the software seamlessly. By clicking on that button, it opens a directory chooser. We can choose the folder which contains the results of the resequencing run. After that, it analyzes the hierarchy of the resequencing folder and shows all the different alignment folders which are present inside the resequencing folder. In this particular case, there happens to be a single alignment folder. After we choose that, it automatically analyzes all the files present in that particular folder, identifies which of them are aligned read files, and those are the BAM files. It looks up the sample naming sheet and identifies that sample 1 actually corresponds to a sample which is RB1. It fills in the proper sample name. Then we just need to click on finish, and all of the data is loaded into Avadis NGS, and a DNA variant analysis experiment is created. The navigator on the left out here shows all the information which has been loaded. We have the reads, and the indel and SNP results have also been loaded into the navigator. The center panel of the tool shows the Genome Browser. By default, the first chromosome is shown, and we can see that the depth is uniformly high across the entire chromosome. The workflow on the right shows all the different QC and analysis steps that can be carried out on DNA data. So now that the results of MiSeq have been loaded into the navigator, into the tool, let us take a look at how the import was carried out. When importing the VCF files which contain the DNA variants, extra columns are added to each variant allele, which allow further downstream filtering and analysis. To be more specific, 4 extra columns are added. For each variant allele, we add a column which indicates the total number of reads which are spanning the allele location in the read file. We also add the percentage of those reads which are supporting the variant allele and the percentage of the reads which are differing from the reference. And finally, since we may wish to filter based on the strand bias, the strand bias column is also added. So all of these 4 columns are added automatically when the VCF files are being imported. Once the data is present inside the tool, we can carry out further downstream analysis. One main filtering step is to identify SNPs which are having an effect on protein function. So Avadis NGS includes a SNP effect analysis workflow step. When we click on that particular workflow step, it asks which of the different effects we wish to identify. By default, it assumes we are interested in identifying all the different SNP effects. When you click on OK, it creates a report where each of the variant alleles has been annotated with the consequence that it has on an overlapping gene or transcript. So you can see out here that the very first variant allele, which is the substitution of G by an A, causes a non-synonymous coding effect in the RSP1435 gene. And to be more precise, that effect happens to occur at the 195th amino acid of the protein sequence and results in a modification of an L to an F amino acid. Similarly, each of the other variant alleles is also annotated with the consequence that it has on the genome. One way of viewing all of these results is through the region list operations utility, which is included with the tool. It allows you to investigate any of those different columns that were shown in the previous sheet. If you notice, there were columns for the total number of reads, the supporting reads, the SNP call, the strand bias, etc. By choosing the channel in this drop down out here, we can check the distribution of that particular values in that column. Here we notice that there are a lot of mutations which result in new Cs and Gs in the genome. So one question one might have at this point is to find out if a specific reference base is mutating into a C and if a specific reference base is mutating into a G. To do that, we can use the powerful visualization capabilities of the region list operation and overlay the color of the reference base. So what we notice now is that of all the bases changing into a C, there are almost equal number of A's, G's and T's which mutated into a C. Similarly, there seem to be an equal number of A's, C's and T's which mutated into a G. So in general, it seems to indicate that there is no particular preference for that mutation. What it indeed shows is that the organism has a very high GC content. It actually has a 70% GC content. So naturally, whatever mutations are happening are resulting in a larger number of C's and G's. So this kind of analysis is possible through the region list operations. One can also try to understand the score that has been generated by the MiSeq SNP analysis software. One would expect the score to be proportional to the total number of reads that is covering the reference location. So we can try to verify that by doing a scatter plot between the total number of reads spanning a reference location and the score that was assigned to the variant allele at that point. And we can see that there are two trends. There are a lot of SNPs for which the score is directly proportional to the total number of reads, but one also sees some outliers out here. Even though the number of reads spanning the reference location is large, the score assigned to that particular variant seems to be quite low. To understand these patterns better, we can color and shape and size the points in the scatter plot by different attributes. So one reason why the score may be low is that the percentage of supporting reads may be low. Similarly, the MiSeq SNP analysis software assesses if the SNP is supported by reads on either strand. So one can also try to plot the strand bias. Once we add all of these features and take a second look at the scatter plot, one will notice that along the diagonal we usually see only the red spots, which indicates that those variants are supported by a large fraction of supporting reads, and also they have very low strand bias, whereas the points along the x-axis out here, you'll notice some of them are disproportionately large and the color is different from red, which indicates that some of them have higher strand biases and some of them have very low supporting reads. We also see some red points out here, and the reason why those alleles got a low score might be because the base qualities of the bases which are supporting the SNP might be lower. To verify those, we'll have to look into the Genome Browser. So the region list operations allows you to understand the data generated by MiSeq better. It also includes a powerful filtering capability. So one can take a look at the spreadsheet of all of the regions that are in the SNP output. One can set different filtering criteria. For instance, one may be interested only in the non-synonymous coding, and we may be interested only in those variant alleles where the total number of reads was at least 100, where the number of supporting reads fraction was at least 40. We can set all these different filtering criteria and observe how many of the regions are going to satisfy this filtering criteria. In this particular case, it is 38 out of 133, and when we click finish, a subset of the region list would be created which contains only the 38 alleles satisfying all these different criteria. After we find out these subset of the variant alleles, we can always drag and drop the region list into the Genome Browser. We can investigate the reads which contributed towards a particular allele being called out. The Genome Browser has a powerful tooltip support. So by placing a mouse on any individual base on any read, we get complete information at the base level and at the read level. Sometimes it's a little bit hard to get an overall perspective in the Genome Browser, especially when the coverage is very high. In this particular case, the coverage is closer to 450. So scrolling through the reads is not going to be an easy operation. So to summarize the information in the Genome Browser, we have a variant support view. By right-clicking on any point in the Genome Browser and choosing the variant support view, we get a 20 base pair window surrounding the chosen location. You can think of it as a vertical slice of the Genome Browser, and all the distinct read sequences have been clustered together. So we can see that there are 264 reads with the first pattern, there are 11 reads with the second pattern, and so on. So we can see at a single glance that there are 264 reads which have an A instead of a T, a C instead of a G, a C instead of a T, and so on. And this indicates that there are correlated SNPs out here, and the SNPs have very high confidence. In this manner, by starting with SNP list, we can find out the effect of the SNPs. We can even invoke the Avadis NGS SNP caller. We can find out commonalities between the SNPs called by Avadis NGS and the SNPs called by the MiSeq run. We can create filtered SNP lists based on different filtering criteria. We can look at the variants in the Genome Browser and assess their significance, etc. In addition, if people are interested, they can directly import the FASTQ file into an alignment experiment, carry out the alignment itself within Avadis NGS before doing the downstream analysis. And all of the operations that are carried out within the tool, they result in objects in the navigator. So you have an audit trail of all the different things that you did to arrive at the conclusions. So let's now take a look at the second popular MiSeq application, which is support for Amplicons. The TrueSeq Amplicon kit, which has recently been released, allows you to carry out high coverage sequencing of up to 384 distinct Amplicons and process 96 samples simultaneously. So if you take a look at a run folder generated by this type of analysis, it would have a huge number of files. You'll notice here that you have 4,000 files in the alignment folder. That is because we are processing 96 distinct samples, and the MiSeq alignment software generates one file for each chromosome. So if you have data for 20 chromosomes, then you already have 100 times 22, close to 2,000 files. In addition, each of the samples has a SNP list and an indel list. So you can see that the number of files quickly adds up, and you have tens of thousands of files in the Amplicon folder. So navigating through these folders and picking up the relevant files for loading into Avadis NGS is clearly going to be a time-consuming operation. So let us see how Avadis NGS helps to navigate the problem. We start by creating a DNA variant analysis experiment. We choose the organism and build against which the reads have been aligned. In this case, it's HG19. We can choose any of the different gene annotations. We provide annotations from Resequencing, UCSC, and Ensemble. Finally, we choose the sequencing platform and library layout. After that, when you're prompted, we click on the MiSeq button and choose the Amplicon folder. In this case, 3 different alignments have been carried out with the FASTQ files in the Amplicon directory. We can choose the appropriate alignment folder, and then all the relevant align read files are automatically populated into the sample loader. You can see that even though the sample number is 1, it has consulted a sample naming sheet and found out that the appropriate name of the sample is NA18502. One can scroll down this list out here and see and verify that the correct sample names have been assigned to these files. Once we click on finish, the data is loaded into a DNA variant analysis experiment. The files loaded are almost exactly the same as the resequencing experiment we discussed earlier. It loads the alignment files, the SNP, and the indel results. The center panel shows Genome Browser with one track for each of the 96 samples. So you can see the scroll bar indicates a lot of aligned read samples are present out there. It shows the dbSNP information and the transcript track. So as we discussed, there are a large number of samples. You can see there are 96 samples, and we do have some replicates. And for each of the samples, we have an indel file and we have a SNP file. But what we'd really like to do is identify SNPs which are common across multiple samples. For that, we first need to create a multi-sample report. At present, we don't create one directly from the MiSeq data, but we will do that shortly. In the meanwhile, if you were to run SNP detection within Avadis NGS, it creates a multi-sample report, which is pretty much a union of the variant alleles observed across all the 96 samples. So any allele observed in any of the 96 samples results in a row in the multi-sample report, which looks like this spreadsheet. So if we take a look at the first row out here, it is a substitution of a C by a G, and that particular variant has been observed in only one sample, and we have data about that particular location for each of the 96 samples. So for each of the 96 samples, we have what is the percentage of supporting reads, the percentage of variant reads, the coverage at that particular location, the strand bias at that location in a particular sample. And if you have 96 such samples, you'll have 96 times 6 columns out here. And the sample count column indicates in how many of the samples has that particular variant been called by the SNP detection software. So you can see that there are some variants which are only specific to a single sample. There are variants which are present in all the 96 samples, etc. One can take a look at the sample count column and see what the distribution looks like. We can see that a vast majority of the variants are specific to only one of the samples, but you do have a small set of variants which are present in all 96 samples. However, we need a robust way of identifying these variants which are present in a large number of samples. So let's try to define what we are after. We are interested in high confidence variant alleles, that is, alleles which have a reasonable amount of coverage in a sample and which have a very low strand bias. Those are what we would call high confidence alleles. In addition, we are interested in identifying high confidence alleles which are common across a lot of the samples. We may also be interested in high confidence alleles which are occurring only in some of the samples. And in the case of cancer sequencing, we may be interested in low frequency mutations. All of these workflow steps are possible through Find Significant SNPs workflow. The first page in the wizard allows you to choose the experimental setup. In this case, we are only analyzing data of 90 different Yoruban samples, and they're all normal samples, so we're going with the first experimental setup. But if we had paired tumor normal samples, then we would go with the second experimental setup. If we had different tissues, we'd go with the third setup, etc. After we choose the experimental setup and move on to the next page, it shows the predefined filtering use cases that are relevant for that setup. By default, the high confidence common variant alleles is the filtering use case which is shown. An allele is considered to be of reasonably high confidence if the percentage of reads which support that allele is at least 35, and an allele is considered to be common if it's present in at least half of the samples. So by default, it is set to half of the total number of samples, which is 48. So what the statement out here shows is that there are 204 alleles which have at least 35% supporting reads in at least 48 of the 96 samples. The chart which is shown out here gives guidelines on how to modify these filtering criteria. For instance, one may be curious on what would happen if the supporting reads were changed from 35 to say 60, and the chart shows that if you were to change from 35 to 60, then the total number of variant alleles that would satisfy the filtering criteria would be close to 72. Similarly, user may be curious on what would happen if the number of supporting samples were changed from 48 to 60, in which case we would consult the colors that are shown out here and say that if you were interested in supporting reads in the 35 to 100 range, but we are interested only in those alleles which have support in at least 60 of the 96 samples, then we would add up these two colors out here and say that the number of alleles would probably be around 170. So the chart gives visual clues on what would happen if these filtering criteria were to change, and the user is free to modify these different criteria. For instance, if we want to set a more stringent value for the percentage supporting reads, we could change it to 85, and now we see that there are only 44 alleles which have such a high confidence in more than 50% of the samples. When we are satisfied with the filtering criteria, we can click on finish, and the 44 different alleles are saved as a region list. We can also carry out clustering on these 44 alleles to see what are the patterns of the SNPs across the 96 samples, and the clustering has been carried out in both the dimensions. So we have clustered the regions and we have clustered the samples out here. Moreover, we also indicated the sample names in the column headers. We have added some grouping information, which I'll come back to in the next slide. So two samples would be next to each other in this particular clustering if they have the same behavior across all the different variant alleles. Similarly, two variant alleles would be next to each other if they have if those alleles show similar behavior across all the different samples. The clustering view, which is new to Avadis NGS 1.3, provides a lot of information. It allows you to customize the colors. In this particular case, the clustering was done based on the percentage of supporting reads. The minimum color blue was set to 0, and the maximum color red was set to 100, with 50% being the yellow color. So let's look at smaller parts of this clustering view to get a better idea of how to interpret this data. The first view shows a part of the sample tree. One can immediately see that samples which cluster very close to each other, for instance NA18912 and NA18912R are the replicates, and you would expect them to cluster next to each other because they should have the same phenomena across all the alleles. Similarly, we can carry out a sanity check and observe that the other pair of replicates also cluster very close to each other. Once we are satisfied with the QC, we can try to see if there are any trends about families. Here we have drawn a particular color bar here where all the samples of the same family are assigned the same color. We get this information from a spreadsheet which shows for each sample what family it belongs to, and this data was from the 30 trios of the European origin. One interesting subset to observe is the pink cluster out here. There are 3 distinct samples, so the entire trio has formed a cluster. You'll notice that the middle member of that particular cluster has a heterozygous version of the allele. The last member of the cluster does not have the allele at all because there are 0% supporting reads, and the first member of the allele of that particular cluster has a homozygous version of that allele. So that would clearly indicate that the center sample is the child sample and the other 2 samples are the father and the mother. One can always redraw the color bar with the family role, the father, mother and child roles to confirm that hypothesis. Similarly, one can look at the region clustering out here, and you'll notice that 2 of the regions seem to cluster very closely to each other. In other words, a sample which has a particular percentage of supporting reads in one region has almost the same percentage of supporting reads in the other region also. If 2 regions are very correlated, one would expect them to be close to each other on the genome, and that is indeed what we find. We see that the 2 locations are only around 95 base pairs apart from each other. We can take a look at the second cluster out here, and we can see that those 2 regions are only 9 base pairs apart on the genome. So one can use the clustering view both as a sanity check and QC. One can also use it to see if there are any interesting patterns that emerge. For instance, if we were to cluster a much more diverse set of samples, one would expect the different ethnicities to cluster closer to each other. And one could then use that as a basis for identifying SNPs which differentiate one population from the other. So all of this information about family and role was loaded through a TSV file which can be imported through the experimental grouping workflow step. Another question people typically have when carrying out Amplicon sequencing is whether all the Amplicons have been covered at sufficient depth in the experiment. For instance, we have 364 different Amplicons. The first quality check to carry out is to see do they have at least 50 reads covering each of the Amplicons in a majority of the samples. So the targeted region QC workflow step in Avadis NGS allows you to take a region list and analyze it, and it annotates each region with the number of reads that fell in that region in a particular sample and the average coverage in that particular sample. So it adds these 2 columns for every sample present in the aligned read list. In our case, we have 96 samples, so there will be 192 additional columns that are added to the target regions. Once we do targeted region QC, we then need to figure out a way of identifying the regions which got low coverage in the majority of the samples. We again perform clustering, where we cluster based on the average coverage, and this time the regions are the columns and the samples are the rows. One can immediately see that the regions in the first block out here have very low coverage in all of the samples, whereas the vast majority of the regions have decent coverage in the majority of the samples. The clustering view also allows you to highlight any individual cluster of interest. Here we have clicked on the first cluster, which seems to contain the majority of the regions which got low coverage. The selected set of regions are shown in greater detail in the bottom panel. One can then save it as a region list, and all these 26 regions which got almost 0 coverage in all of the samples will be saved as a region list. One can then look into where the reasons for the low coverage were. It could be that there is something wrong with the Amplicons. It could be that these regions are deleted in these samples. Another way of identifying these regions is to run the find significance region script, which is packaged with the tool. We can choose the numeric column based on which we want to filter the regions. So we want to filter based on average coverage, and we're interested in finding out which are the regions which have low average coverage in the majority of the samples. After specifying this filter criteria, we see that 26 out of the 363 target regions have low coverage. We can save these regions as a region list, and in this particular case, they happen to almost exactly match the results of the clustering view. So in this way, Avadis NGS helps you to filter through the large number of samples and SNPs that are generated by the MiSeq analysis software. We now move on to small RNA analysis. The MiSeq software quantifies the small RNA analysis results in the form of a file called hits.txt. The first column of the file gives the identifier of a mature miRNA. All of the other columns indicate the number of reads that align against that particular mature miRNA in each one of the samples. So you can see there are 4 samples out here labeled 1, 2, 3, 4, and HSA LED7A. There are 23,000 reads that align against it in sample 1. There are 26,000 reads which align against it in sample 2, etc. So this spreadsheet could be used as a basis for differential expression, but Avadis NGS actually provides a lot more capabilities regarding small RNA analysis. It allows for quantification of other small RNA species such as tRNAs, snRNAs, snRNAs, etc. It helps to discover novel small RNA genes which are not present in the annotations. It also helps to identify annotation discrepancies. But to perform all of these analyses, we need to align the reads against the entire genome, and that is possible through a small RNA alignment experiment. So if we create a small RNA alignment experiment and we click on the MiSeq button, we can choose the small RNA folder. It automatically looks through the folder, identifies the FASTA-Q files which are present inside the folder, and shows the sample loading sheet. It consults a sample sheet file to find the sample name to associate it with each one of those sample numbers. It goes through the FASTA-Q files. It drops off the reads which have been flagged as invalid by the Illumina software. It carries out the demultiplexing and the sample naming. And once the samples are loaded into the navigator, one can carry out an alignment against the reference genome. And after the alignment is completed, one can launch the small RNA analysis experiment. The details of small RNA analysis were covered in a different webinar, so I will not be going into those details today. So I've covered so far the 3 different types of experiments for which we provide full-fledged support within Avadis NGS. And so those were resequencing, Amplicon sequencing, and small RNA analysis. I'll show a few slides about the other MiSeq application areas for which we have experimented internally with how to provide support, but have not included any support within the tool as such. Customers who are interested in seeing these features within Avadis NGS, please get in touch with us, send us your comments, and we'll try to see how to incorporate these features in future versions. We start with 16S rRNA metagenomics. What we can do is create a reference which contains all the different 16S rRNA genes of interest. The reference can be loaded into the Avadis NGS software through the annotations manager. And because the aligner is built into the software, we can now align all the metagenomics reads against this new reference that has been created, and the results can be visualized in the Genome Browser. So here we show all the distinct 16S rRNA genes labeled by the organism name. We can look at the number of reads which have been aligned against each of these genes, and that serves as a quantification of that particular bacterial species. Each of the bacterial species also has a taxonomy tree associated with it. So we know the genus, the family, the phyla, etc. So what one would want to do next is to explore the data at different levels of this particular taxonomy tree. One could try to display the results in a traditional tree structure. The tree structure is good for exploring the taxonomy, but it is hard to indicate the relative proportions of the reads that align against different members of the taxonomy. So one way of showing the proportional representation is via a drillable pie chart. So the pie chart, the individual pies indicate the fraction of reads that align against that particular bacteria. But in addition, one can double click on the pie, it will show the next level of the taxonomy. This is one experiment we are carrying out with how to explore the metagenomics data in an intuitive manner. We've also done some preliminary work on viewing de novo assembly results. The MiSeq software uses Velvet to carry out de novo assembly. The result of the assembly is a set of contigs and reads which align against those contigs. We have used the AMOS tool set to convert the alignment results into SAM format. Once we have the alignments against the contigs in SAM format, we can load all the different contigs as different pseudochromosomes into the Genome Browser. We can also load the SAM files to observe what is the depth of coverage at different points of the contig, to observe whether the paired reads which are aligned against this contig, do they support the insert length distributions that we expect, and so on. So here you can see that the first contig had length 17 kb. The coverage is reasonably high across most of the contig, but we do see certain discrepancies. We can see certain locations where the consensus sequence doesn't really agree with the contig. And as a sanity check, we would be interested in seeing that the reads which are towards the end of the contig have mates which fall in other contigs. That could be a basis for joining these contigs into a scaffold, but those are visualizations which are not explored right now. So based on customer feedback, we will see how to incorporate support for these additional MiSeq applications into future versions of Avadis NGS. So that brings me to an end of the webinar.